Good afternoon, everyone. My name is Olga Shavuka. I am a lab scientist for the Group for Research in Infectious Diseases team at the University of Namibia. And today I'll be presenting on a study that is titled The Population Structure and Transmission Analysis of Drug-Resistant Tuberculosis Strains from Namibia that were analyzed using whole genome sequencing. I have nothing to disclose. Okay, so Namibia is the 34th largest country in terms of surface area, but we have one of the lowest populations. The population was estimated to be 3 million in 2023, yet we were ranked as having the 30th highest TB burden by the World Health Organization. The TB incidence for that year was 468 per 100,000 people, and that totaled to about 14,000 cases, and more than a quarter of those were people living with HIV. Here, the graph shows the annual notification of confirmed drug-resistant tuberculosis cases. There was a decline during the COVID period, as you can see. The cases confirmed were about 213, but experts predicted that after the pandemic, the cases will gradually start to increase, as you can see in the graph. And in 2023, the confirmed number of drug-resistant tuberculosis cases was 311, but this was significantly lower than the World Health Organization's estimated number of 630. This means that we are not finding and diagnosing more than 50% of all drug-resistant tuberculosis cases in our country. So this graph shows the notified MDR-TB cases for 2023. And even though Namibia is missing so many cases, the numbers notified are still high enough that we are not doing so great when you compare us to other countries, as you can see by the intensity of the green when you compare us to many other countries. So what we did was include all the samples from patients that were rifampicin resistant according to the gene expert, and that had a positive culture. These were samples that were collected between 2016 and 2023. DNA or CTAB extraction was then done at the University of Namibia, and the samples were shipped to Research Center Bostel in Germany, where the library preparation was done. Using the Nextera library preparation kit, and the quality control was done using a fragment analyzer. The samples were then sequenced, and then the FASTQ files were uploaded into SexSphere, where the cluster analysis was done using a threshold of 12 alleles. Okay, these are the results. So about 525 samples underwent whole genome sequencing, and as you can see in the picture, they were classified as being either sensitive, multi-drug resistant, non-multi-drug resistant, pre-extensively drug resistant, and extensively drug resistant. And as you can see in the map, that most of the regions were predominantly multi-drug resistant, as you can see by the reddish dominating color on the map. Okay, so the cluster rate was high, with about 70% of the isolates being grouped into 78 clusters. About 76% of the MDR strains were clustered in were grouped into 61 clusters, with isolates ranging from 2 to 76 isolates. And then about 82% of the pre-XDR strains were clustered or were grouped into 9 clusters. And then 100% of the XDR strains were grouped into 4 clusters. So the presence of clusters does show that transmission plays a huge role in driving drug-resistant tuberculosis in Namibia, and it is quite concerning that these dangerous strains such as pre-XDR and XDR have clustered like this. This means that these dangerous strains are really circulating in our communities. About 97% of the strains were classified as being lineage 4, 2% were classified as being lineage 2, and then 1% was classified as being lineage 1 and lineage 3. This is the same minimum spanning tree, but this one is colored by sublineage. And as you can see by the light pink, or no, light blue color, so the largest clusters were all the lamb sublineage. And the lineage 4 dominance aligns with findings from other African countries such as Eswatini and Zambia. So our results have shown that transmission actually plays a larger role than acquisition of resistance in driving drug-resistant tuberculosis in our country. Further research is needed to investigate the potential drivers because only by addressing these drivers is key to controlling the spread of drug-resistant tuberculosis in our country. Okay, these are my references. This is a picture of our group. And these are our collaborators. Thank you very much.